So our first speaker of this session is Jaco Visser. His talk is a trait-based framework to understand and predict the response of wild bee functional groups to anthropogenic landscapes.
Great, thank you very much. I hope you can all hear me well.
Yes.
My name is Jaco Visser. I'm a PhD student at Charles University in Prague in the Czech Republic and at the Free University in Brussels in Belgium. And like Catherine said, I'm here to talk about our recently published paper, a trait-based framework to understand and predict the response of wild bee functional group to anthropogenic landscapes. A small overview of what I'll talk about. Our goal in this study was to create functional groups based on a very large data set with many species and to have a complete database for all of those species. Then we wanted to do landscape classification as broad as possible, but still equally relevant. And then to combine these two to see how these functional groups are associated with certain land use types and certain landscape compositions. So first I'll talk about the data set. I sampled in the Czech Republic, which is in Central Europe, in the two main cities of the country, Prague and Brno. There we selected 32 locations, 16 per city. Half of those were in agricultural landscapes and the other half within urban landscapes. And then we calculated the percentage of suitable bee habitat cover, which we designated to be grassland cover, tree cover, and the cover of small woody features like hedgerows and shrubs. And we did that in a 1 kilometer radius buffer zone around the sampling site. So on the right, you can see how we combine the three main habitat types to create this map of suitable bee habitat for each of these locations. So in total, 32 locations. We then sampled for 3 years with pan traps. Each month we sampled for 5 days on each of the locations. I then pinned almost all of the bees, which was just over 26,000 specimens. I pinned almost all of them, and then I identified them all to species level, which resulted in 283 species of bees, which is quite good because in the Czech Republic there are about 600 species. We then combined that by creating a very large trait database for each of those species. And the trait database consisted of 3 nesting traits: nesting type, nesting strategy, so do they excavate their nest, do they rent an existing cavity, etc. And then how do they respond to other bees during their nesting, in other words, the sociality. Then foraging traits, the tongue length, which determines which kind of nectar the bees can which kind of flowers they can visit for nectar, their pollen specialization, how they transport their pollen. And then we have 2 traits, which are the interaction between nesting and foraging, the intertegular distance, which is a proxy for how far the bees will forage, and then seasonality, when do the bees fly. So with these 2 databases in hand, we wanted to create functional groups. And before I show you the results of this, I find it important to highlight that in bee studies, in functional trait studies of wild bees, the term functional group is used a lot, but technically almost always incorrectly. What you will find in wild bee studies on functional traits is where a single trait is treated as a group. For example, all kleptoparasites are seen as one functional group, or all oligolectic species are seen as one functional group. But of course, this runs into problems because if all kleptoparasites are one functional group, all the species within that group still vary in all of the other traits. So you're missing a lot of ecologically relevant nuance there. So we took inspiration from studies into other taxa, and we used different clustering methods to group the bees together based on all of their traits. By doing so, we found 18 distinct functional groups for our data set, and you can see all the splits there. We have splits from parasitic to non-parasitic, polylactic, oligolectic, species that nest in the soil or in cavities, spring active, summer active, etc., etc. And by combining all of those, we got these 18 functional groups. And there already we had an important result. What I just mentioned, grouping all, for example, oligolectic species together in one functional trait, would be wrong in our case, because with our clustering we showed that oligolectic species in our data set are actually three different functional groups. We have the cavity nesters and the soil nesters, and then the soil nesters are split between spring active and summer active species. And the same pattern could be seen with the kleptoparasites. First of all, social parasitic bumblebees are not kleptoparasites, so they are also a distinct functional group, number 13 in this dendrogram. And then our kleptoparasites will actually split into four functional groups. We have our summer active and spring active, we have our cavity versus soil nesters, and we have polylactic and oligolectic kleptoparasites. So with our functional groups in hand, we then wanted to see how are these functional groups associated with the landscape in which we found them. Multi-species distribution model, where you can see all of the important traits for the different functional groups. You see the 18 functional groups there. And then in red are negative association with that land use type, and in blue are the positive associations with the land use type. So you can see that there's a lot of variation between the functional groups, which land use type they are associated with, both urban and agricultural, but also the three habitat types. And then I would like to draw your attention again to the grouping of the oligolectic species in this case. Grouping them all together was, in our case, wrong from a clustering perspective, but also from an ecological perspective, because we can see that these three groups are associated with very different habitat compositions. And the same can be said for our kleptoparasites. There is no clear land use type which all of them are associated with, so grouping them together would be a mistake. And then finally, another final example would be the cavity nesters. We have four groups of cavity nesters, one in the kleptoparasites, one in the oligolectic species, and then we have cavity renters and cavity excavators. Of course, because they are all cavity nesters, they are all positively associated with tree cover because they need cavities to nest in, but they require very different complementing habitats. So also grouping all cavity nesters, for example, together, or all soil nesters together, is an incorrect use of functional groups in this case. So we then saw how the different functional groups are associated with the different land use types. And then we wanted to see if the landscape changes, if the landscape composition changes in a certain way that is beneficial for one group, is it also beneficial for another group? So in this heat map, you can see a correlation. So red again is a negative correlation and blue is a positive correlation. And you can see if certain functional groups, if the landscape changes that in a beneficial way for them, if the other group responds similarly or oppositely to those changes. I'll give you an example. So functional group 4 are the Hileus species. So these are typical stem nesters, very small bees. And group number 12 here are the obligatorily eusocial species. So you can see that if the landscape changes in a way that is positive for functional group 4, it is also positive for functional group 12, our eusocial species. So this, of course, is important from a landscape restoration point of view, because if you have a landscape that you're either converting, say, from agricultural to nature, and you want to aim your restoration at bees, you need to know which type of landscapes you need to create. And it's also important to know which species, or in this case, which functional groups, are positively or negatively influenced by your decisions. But then we went a step further, because we realized that there is a difference in this correlation when you split the locations between urban on the left and agricultural landscapes on the right. And we can see when we look at the same two groups, the functional group 4 and 12, that their association or their correlation is always positive in both environments. However, it's much stronger in the urban environment. But this is not with all functional groups. For example, if we keep the Hileus of functional group 4 and we compare it to functional group 7, which mainly has Anthidium species and Anthophora species, we can see that in the urban environment, when the landscape changes in a way that's beneficial for one group, then it is detrimental for the other group, so they have an opposite reaction. However, this correlation does not exist at all in the agricultural landscape. And maybe even more importantly, when we keep functional group 10 and we compare it to, when we compare functional group 10 to functional group 7, so again, functional group 7 are the Anthidium species and Anthophora species, while the other group here are mainly Andrena species, so from a trait-based point of view, very different species. We see that in the urban environments, they respond oppositely to changes in the landscape composition, while in the agricultural environment, they respond similarly. And this is important to know because if you're restoring a urban park or you're restoring some kind of agricultural landscape, you need to know the nuances in which functional groups or which species you are benefiting or not, and that the matrix in between, so the anthropogenic matrix in between your habitats, does make a difference with these in these cases. Some concluding remarks then. So first of all, we tried to see what the difference was between using functional groups and functional traits. In the end, functional groups are very useful because they capture the full trait variety within a species, not just one trait or focusing on one trait. And we really emphasize that using a single trait as a quote-unquote functional group is an incorrect way of using the term functional group. Then functional groups have clear associations with, in our case, very broad landscape types. And functional groups can react the same or oppositely to changes in the landscape compared to other functional groups. And of course, this is important, like I said, for conservation and restoration efforts. And finally, like I showed in the last figure, that the anthropogenic matrix, so agricultural or urban, between the habitat patches matters because bees respond differently, what kind of anthropogenic matrix they have to fly over or across to get that to their habitats. All right, thank you very much for listening. I'm happy to answer any questions you have.